Dr. Wells has supervised and graduated 14 master and PhD graduate students, and currently he has 2 PhD and 4 master students in progress. For professional experience, Dr. Wells was the president for North American Forensic Entomology Association. He is also an editorial board member in the Journal of Forensic Science Institute, and he has served as an expert consultant for more than 50 criminal cases in Alabama, Arizona, California, to name a few. And Dr. Wells' research concerned about the development of new genotyping methods and statistical analysis for forensic biology and insect evolution. His current research project include the novel protocols for human identity and paternity testing, population genetics of forensically important insects, biosystematics of the fly superfamily Ostracidea, and statistical methods for estimating the time of death. So without further ado, let us welcome Dr. Wells to share his talk with us, the logic of forensic entomology analysis and associating a probability with an estimate of postmortem interval. The floor is yours, Dr. Wells.
Thank you so much, Dr. Chin. And it is a great honor to have been invited to this institute to meet such interesting and kind people and the wonderful country of Malaysia. I hope I can repay the favor through future collaborations and academic exchange. And thank you all for coming. I'm going to start and end this talk with a case study. And let's click on, do the arrows work on this? There you go, okay. And I am actually asking, it's difficult for me to get questions from people who are viewing remotely, but I will try. How would you interpret this case? A case in which there are no insects involved. I'll go through it relatively quickly. And if you could mute your microphone, the people watching remotely. So this occurred in the United States city of Nevada, Las Vegas, which you probably know. It is a very urban location within a desert part of the country. The exact location was right in the center of town. I'll show you ground level views shortly, but just to show you that the relative position of where the victim was found by a homeless person, who then walked over and used a telephone at this convenience store to call the police. And I think probably that didn't take very long for that journey. And so the basic timeline is that call to police took place at 10:36 p.m. In July, so it's very warm. Police, of course, arrived immediately, and then the coroner's team arrived early the following morning and observed that at about 4 a.m., the corpse at that point was in full rigor. Sent to autopsy, where it formally confirmed what was obvious already. This was a vicious murder. Death was caused by blunt force trauma to the head. There were, as I recall, 18 stab wounds and extensive mutilation post-mortem, including severing the penis. Location was this parking lot. This is behind a bank at the time, and you see this masonry wall. This is a enclosure for what we call a dumpster, a large rubbish container. And this, because this was not a household, there wasn't food waste. There was not the kind of trash that would attract insects. The material you see behind the dumpster had been pulled out to cover the victim. And what you see is the head there and the foot there. I'm not going to show you the actual corpse, but what's quite important is that when all of this was removed, you see this massive amount of blood. Blood pooled, blood spattered. And so I ask you, I'm really asking you, the people who are listening right now, where do you think the corpse was between death and discovery? Don't overthink this. Right, exactly, right here. Death occurred right at that spot. And I'll discuss why I emphasize that in a bit. In other words, if you were able to infer the amount of time the corpse had been at that location, you would be inferring postmortem interval. As I said, this is a no insect case. There were no forensic entomologists on the scene, but there was extensive examination of many photographs, and the autopsy didn't report any carrion insect. It appeared that there were no fly eggs or larvae, not even any cockroaches on the corpse. The weather was fine. I didn't emphasize it, the photographs, but there was no barrier to insect access. It was easy for an insect to reach the corpse, and no barrier to the escape of odors. So let me ask you again, from an entomological perspective, what would you infer about how long this person has been dead? It could be in very general terms. Everyone's wearing masks, so I can't. Say it again, please. So yes, not very long. You could get more precise, for example, based on the rider at 4 a.m. But the point is, you can confidently say not dead for very long. And the time of death was actually quite important here. I've simplified this. This wasn't exactly the case, but the situation was, if death occurred after sunset, that would be very favorable to the suspect. 7 forensic entomologists consulted on this case. And most of them reached some sort of conclusion that if that corpse had been present at that location during daylight, prior to sunset that evening, if that had been true, there would have been eggs on that corpse. I didn't agree with that exact conclusion, that any amount of daylight at all would have been enough time for flies to find and lay eggs in the corpse. But I agree with the general logic that one can interpret the lack of insects on a corpse if conditions are suitable for insect activity. One can interpret the lack of insects on the corpse as some kind of maximum time since death, dead for not more than a certain amount of time. And again, I'm kind of belaboring the point here, but I'll just go right ahead. And it's been my impression with the forensic entomological community that almost anybody would reach some similar conclusion, given a corpse exposed to potential insect activity under environmental conditions that are suitable for insect activity. Almost anybody would be willing to reject some hypothesis of time since death, whatever it is. Could this person have been dead for 2 hours and still not have been colonized? You might say yes. But if I say, okay, could it have been 1 day? Could it have been 2 days? At some point, I will reach a postmortem interval where you will say, no, it couldn't have been that long. The corpse could not have been there for 3 days and still not have been colonized. I'm belaboring this point because when you do that kind of logic, you're estimating maximum postmortem interval based on insect biology. And if you are willing to do that, you are voicing an opinion that is incompatible, logically incompatible with a common statement in the forensic entomological literature, and also common statements I've heard from forensic entomologists, namely that all a forensic entomologist can do is estimate minimum postmortem interval, never actual postmortem interval. And something I want to emphasize here is that is not true. Let me get into some of the reasons offered in support of the claim that a forensic entomological analysis can only yield sometimes a minimum postmortem interval. So some people will say, well, the history of the corpse is unknowable. You can't estimate postmortem interval because the corpse, the victim may have been killed somewhere else. Insects couldn't get to it and then it was moved there later. And as I've just demonstrated, sometimes that is not true. You can know that the victim was killed right there and has been at that spot until discovery. People will claim that the pre-appearance interval, this is the time from when the corpse becomes potentially available to insects to when it is colonized. They'll say that you can't know that. But that's not true. Also, you can measure that. That's something you can experimentally measure. And in a related point, people will say you can't estimate postmortem interval because the way you estimate postmortem interval is just a development analysis. You estimate the age of a maggot. You find the oldest maggot on the corpse that you can, and then you say, okay, if this is 4 days old, then the person has been dead for 4 days. And people object to that because of the previous bullet point saying that you cannot know the time between death and when the mother of that individual larva was deposited upon the corpse. But that kind of PMI analysis based on development, that's not a serious method. If you look at the literature on how to estimate PMI using forensic entomology knowledge, people don't propose that. They propose it as a succession analysis. So people who argue in this way are not dealing with the actual way people have said you can estimate PMI. And there is a literature on that. There has been for many years. And papers by people who are not obscure, like Gail Anderson or Jens Ament or Martin Villett or me, if you care what I say. So there's a substantial literature out there saying that you can estimate PMI. And the people who claim that you cannot never deal with those papers. They never say, because I disagree with those papers, they just don't address them at all. So I'll end this rant by saying the claim that a forensic entomological analysis can never, I'm not saying in every case, but can never yield postmortem interval is contradicted by logic. It's contradicted by practice because forensic entomologists do estimate postmortem interval and they do testify in court, at least in North America and Europe. And there is a substantial published literature describing how it can be done. So please, let's stop saying that a forensic entomologist cannot estimate PMI and can only estimate minimum PMI. It's not true. So after that bombastic interval, let me just review the field in general. I think most of you are familiar with this. We rely upon two postmortem clocks. One is a development analysis, that is, you estimate the age of an immature insect, and I'll just mention this can only be done one specimen at a time. Or you estimate stage of succession. And by stage, I'm talking about the combination of species found on the corpse. And this changes with time as decomposition proceeds. I'm not referring to the morphological stages of decomposition. You hear about fresh, bloat, decay, whatever it is. Although you often see a statement that the insect succession is related to that. It is not at all. There's no set of insects that are characteristic of the fresh stage and that are distinct from the insects that are found in the bloat stage. There's no connection at all. And if we didn't pay attention to those physical stages to decomposition, it wouldn't hurt us in any way. It wouldn't affect forensic entomological analysis at all. And when I say age, of course, that's not just time. It also has to be adjusted for temperature. I can't see what it says. Okay, so that just says simply. I'm going to go into the logic of doing a development or succession analysis, and this is overly simple, but it gives the idea. So when you estimate age, this is usually done with reference to one of the many growth studies. The literature is full of maggot growth studies, and people have examined all kinds of species and all kinds of temperatures and cyclic temperatures and constant temperatures and the effect of various concentrations of drugs. And so faced with an insect of unknown age, you will typically go to the literature and find a reference data set that you think is appropriate for your crime. Now, people have been very concerned with what makes the data appropriate for your particular investigation. Is it the right species? Is it the right temperature? Does it take account of whatever drugs might be involved? These are very important considerations, but that's not what I'm talking about today. What I'm going to be talking about today is how you should proceed once you decide you have the appropriate data to proceed. Okay. And let's say this, you can't see this, I don't think, but this is Megacelia abdita. So it's a phorid, and the lowest line is 10 degrees C. So let's imagine you thought that was appropriate. And so if you have a larva that size, it's that old. Okay, that's a little too simple, but that's basically what the logic is. And in the case of, and that can be interpreted as a minimum postmortem interval because this is a development analysis, and it does not include any estimate of the time elapsed from death to when that individual was deposited on the corpse. That would be a succession analysis. And then succession, again, we could potentially take abundance into consideration because that's probably important, but the methods have not been well worked out for that. For succession, we analyze occurrence, which is presence and absence. And again, in this very simple case, you've done some succession experiment, you've put out pigs, you put out humans, and you come up with a succession pattern. And those that are present or absent as a function of time since death. And if you have what I call a mystery corpse, you know the combination of species on it, but you don't know how long it's been dead. And that mystery corpse has that combination of species, which only, in this case, were only found on day 3, then you would say dead for 3 days. And that is an actual PMI. Okay? It's not, there's not a way that I know of that you can use succession analysis to come up with a minimum PMI. It doesn't make sense, given the data we have. So to make that interpretation, to interpret the age of an immature insect as a minimum PMI, you're making the assumption that that individual got to the corpse by being deposited by its mother on or near the victim following death. That does not have to be the case. People are infested while they are alive. And we know of cases where someone was infested while they are alive and then died. So in that case, the larva was present before death, so its age is not a minimum postmortem interval. It is also possible the larva could crawl to the corpse from some other corpse or other decaying material. In that case, time of colonization is not age. Okay? So in that case, it would not be appropriate to interpret the age as a minimum postmortem interval. These, however, are rare circumstances and can usually be inferred from the crime scene. So it's not, I think, much of a problem. And in the case of succession analysis, this is interpreted as PMI if the victim died where she or he was discovered. And that's because of the kind of succession experiments we do where we mimic death at the scene. And again, you must have suitable reference data, what we call training data. But I'm starting with the point in the investigation where you've decided that you have that, and how should you proceed? So I realized that my presentation was full of boring slides that were just text and diagrams and didn't have very many pretty pictures. So I thought I should at least pause for a second and remind you that carrion insects are beautiful. And I see Dr. Karahashi here today, and he's the one who taught me how to do a species description. And the larvae are exciting. Now back to the boring stuff. So I talked earlier about the basic logic of estimating maggot age or succession interval. It's a bit more complicated than that, but when I say we, I mean mostly my longtime colleague, Dr. Lynn Lamont of Louisiana State University, who is the mathematical and statistical brain behind our work. We believe that it could be better. And we believe it could be better in the following ways. We think that any conclusion should be accompanied by an objective statement of uncertainty so that the police or the courts can decide for themselves how much weight to give to your evidence. We believe that having methods for coming up with that objective statement of uncertainty would greatly improve how we do experiments. I think that an estimate of postmortem interval or insect age should be a range rather than a point estimate. And that one suitable way to define that range would be as a statistical confidence set. This is a mainstream approach. This is how people do it in many branches of science. There may be other ways you want to do it. Maybe you're a Bayesian. Maybe you want to do some kind of machine learning thing. There are other ways to do this, but one suitable way to do this would be as a confidence set. And if we did this, this would improve casework, but also, as I alluded to earlier, it would improve how we do experiments, and it would make possible solid validation experiments. So I think you all understand what I mean in terms of empirical validation. This is a performance test of your analytical method. So in the case of, let's take the simple example, you have a method for predicting, that's what we say in statistics, we predict the age of a maggot. And if it's a validation study, then somebody must know the correct answer and see whether you get the correct answer or not. So let's say you know that the maggot is 4.5 days old. It would be unrealistic to say that your method is only good if you predict exactly 4.5 days. That's expecting too much. So how close is good enough? The confidence set tells you how close. The confidence set includes the true value, then you've made a correct prediction. And I'll get into this later, but for these kinds of prediction statistics where you are making a prediction in the form of a confidence set, the standard performance measure in statistical science, widely used and for many years, is something called the coverage. And I will describe that more in a later slide. So again, my whole point is there may be other ways to do this, but this is an acceptable mainstream way to approach this problem. So before all of you were born, we began to work on this by adapting a classical branch of statistics called inverse prediction, which has been around since the 1960s. Inverse prediction is a method for predicting what we call predicting condition from response. So in the case of maggot age, the condition would be age, the response would be whatever it is you measure on the insect, or the instar size or something. It could be gene expression, something like that. In the setting of a regression analysis, it would be predicting X from Y. And we did this because this is well established, and coming up with a statistical analysis that you can trust in a wide variety of situations is not easy. It takes a lot of work. It takes a lot of work to investigate the mathematical properties, the probability statements, whether the test is robust to small deviations from the assumptions, whether it's reliable. And that takes a lot of work, and this has already been done for inverse prediction. So it was useful to start from that starting point. And also it is appropriate for this kind of hypothesis. It is designed for predicting condition from response, and that's what we want to do in forensic entomology. And if you pick up a standard textbook and look near the back, you'll probably find a chapter that says something like predicting X from Y. And it is done, as it has been for many years, within the context of model one linear regression. And as is the case often with parametric statistics. Your estimate is not directly made. It's what you do is reject some values. So it's given a y, you reject some x values as being untenable. They couldn't be associated with that y. And those values that you do not reject form your confidence set. So this appeared to be promising, but it was not appropriate for forensic entomology data. For example, this is based on data that are linear. These are based on data that are homoscedastic, meaning the variance doesn't change with X, and that is not the case with, for example, maggot growth data. They are not nonlinear, they are heteroscedastic. So you couldn't just apply classical inverse prediction to them. And also succession data are categorical, and I'll get into these tables later. And there was no method for doing inverse prediction based on categorical data. Relatively early on, we solved both these problems, and by we, I mean mostly Lynn Lamont. I fed him the data. I told him what was important to the forensic investigators, but he was the mathematical mind behind this. So we adapted it to nonlinear and heteroscedastic data and invented inverse prediction for categorical data, which is an entirely new statistical test. It did not exist before then. And we kept working on this. I can get this to go away. Okay, let me make that. Thank you. All right. Please excuse my computer illiteracy. So anyway, we did a whole, I can't remember how many papers, 6 or 8 on this, and the methods are all worked out, right? And this is the most comprehensive paper in which it shows how to do a multivariate analysis. You can do a multivariate response. It can include continuous quantitative and categorical data in the same analysis, and it can be implemented in standard statistical software, which was not the case before. This is one of the reasons people didn't do inverse prediction, was you had to program yourself. So it's available now. It's available to anyone. If you want to take this approach, you can. It can be done. Well, what do I mean, if you want to do it? Well, I have to say that actually I can't do it. I can't do the multivariate analysis myself. And I doubt that most of you can either, but you can do it with the assistance of a statistician. And you should form an association with a statistician, a highly trained person, someone with a PhD in statistics or mathematics. And we wrote the statistical section of our PMI chapter in the 3rd edition of Forensic Entomology, specifically for the forensic entomologist to take that text to the statistician, so that the statistician would understand the setting and the statistical issues and would be able to help you. That was the purpose of that part of that chapter. However, I can do a univariate analysis myself. I can do a univariate continuous variable, and the categorical data could not be easier. The categorical inverse prediction doesn't require any calculation at all. You simply consult a table. So anyone could do that. And I'll give you an example of this. So categorical data, again, would be presence or absence in a succession experiment, or life stage in a development experiment. And so you come up with a table, and you do a series of contingency table comparisons. And let me take you through this. So let's imagine estimating PMI from succession. And this is just a theoretical example. So you have a mystery corpse. I'll use that term, mystery corpse or mystery maggot, in which you know the response. In this case, you know the species combination, which is both A and B present, but you don't know the condition. You don't know PMI. And you think you have some suitable reference data, which I'm going to use the same table that I showed on an earlier slide. And so in this theoretical example, what you've done a succession experiment with pigs or human corpses, and you use 10. and you left it out in this case for 7 days. On each of those 7 days, you recorded the number of those 10 that had a particular species combination. So in this case, the apostrophe means absent. So the top row is A present, B absent. So on day 1, all 10 replicate experimental corpses had A present, B absent, etc. And so you've got a mystery corpse that has A present, B present. And so you compare that to the training data 1 day at a time. You compare it for each day. You make a contingency table comparison, which I'll show you in a second, and you reject or fail to reject the hypothesis that that is the PMI. How does that work? Let's look at day 1. It's a strange contingency table. That left column, which TD is training data. You notice that those are the same values as the left column in the table. And then what's really odd is the mystery carcass. This is what makes this a strange contingency table. It just has a single value in one cell. The reason a new statistical test had to be developed for this is because classical methods like a chi-square or an exact test don't work. They give incorrect p-values for a contingency table when you have only one value there. But let's forget the statistics for a moment and just think about how you would interpret this. So you've got a mystery carcass that has, or corpse that has A present, B present, and you're thinking, could it be 1 day old? And you decided that that table of data on the left is appropriate. And you'd probably say, no, no corpses had A and B both present on day 1. So I don't think day 1 is a tenable estimate of time since death. I'm going to reject that. But how about day 3? Well, yeah, it could be day 3. All of the experimental corpses had that combination on day 3. So I wouldn't reject that. Okay? So that's the non-statistical approach. And it's all based on the value in this cell, the training data cell, adjacent to where your mystery corpse 1 value is. Is this value low enough that you would reject the hypothesis that that is part of your confidence set? In these 2 cases, it's clear, but sometimes it won't be so clear. It won't be all or nothing. So how do you decide that that value is low enough to reject? Well, we tell you. We just put a table in our publications, and you just look at the table and decide and see the critical value, this value or lower, you reject. Higher, you don't reject. And again, the non-rejected values form your prediction. And it's probably a prediction interval. We say confidence set. A confidence interval is one kind of confidence set, and hopefully you'll get a continuous confidence set. So I'm going to show a couple of references here. If you want further examples of how to do this, I don't mean for you all to read this right now. We're recording this presentation, and so if you want to know more and see examples, please look at the video and read these papers, which will take you through this process. Okay? So that's how easy it is. You don't even need a calculator. You can just look at the table. You just compile your data, look at the table, and you can come up with a confidence set for a prediction of PMI for a mystery corpse that is a particular combination of taxa. So now that you can do this, as I mentioned before, this provides an objective standard for method validation. And so if you're making, for example, 95% prediction intervals, you're predicting with 95% confidence, and you understand the condition response relationship, then 0.95 or better of your predictions should be correct. Now, that doesn't mean you would give up if it were lower than that. Let's say you did a performance evaluation and your accuracy was 0.85. That may be good enough for you. And of course, you would report that to the police or to the legal system, so they would decide if that was good enough for them. But if it was less than 0.95, if that was your level of significance, then you know you had work to do to better understand the condition response relationship. And once you if you were to adopt this prediction-centered approach to forensic entomology, it would have implications for how you design your experiments. And again, training data is reference data. And this is important because I'm sure you realize forensic entomology experiments are very labor intensive. And because of that, people sacrifice replication because they just don't have time to do more. So how would we optimize our experiments so that we could do more replication, for example? Well, one way would be to identify measurement variables that don't help you. Okay? They do not promote prediction performance, so don't include them in your experiment. And I'm going to give you an example of how this could work. I'm going to show you a table here. And so let's imagine, and in this case, we did a simple growth experiment. This is Chrysomya megacephala, reared at 27 degrees and measured length, width, and instar of each larva. And then used those measurements to make inverse prediction functions based on every combination of those measurement variables. So just on length, just on width, just on instar, and then various combinations of the two to see how that would affect the performance of the prediction. And this was in the example I'm going to give you, this is just going to be the prediction of a single larva, age of a single larva that we know is 56 hours old. So take a look at this table. So across the top is the sample ages, 17, 8 hours to whatever it is on the right. And then each row is a prediction given a particular statistical model. The top row is just based on length, etc. The rejected ages are the asterisks. So the prediction is the gap. Okay, the gap is your prediction of age for that larva, and as if you didn't know how old it was. And first thing you might notice is that all the predictions are accurate in that they all capture 56 hours, but they're not the same width. And you would rather have a narrow prediction compared to a wide prediction. You would rather have a prediction that did not include incorrect values. So you see, as you move from the top row down, you add variables to the model, the prediction becomes better in that it becomes more narrow. But in that final step between a prediction based on width and instar to one based on length, width, and instar, you don't gain any performance. If you've measured width and instar, measuring length doesn't do you any good. So if you did this in a preliminary experiment, you might decide, well, but then I scale this up to a large rearing experiment where you're doing tens of thousands of individuals, which is what you should be doing if you're doing a development study for reference data. You won't measure length, which would be a tremendous time saving if you're measuring thousands of larvae, and would allow you to increase the size of your experiment. We also early on explored the effective sampling method. And again, what I'm talking about here is looking at how you sample your data and what are the implications for prediction performance, because that's what you want to optimize, is prediction performance. And this example is becoming a little dated because people don't do this much anymore. But not that long ago, people were doing a rearing experiment. They say that you wanted to look at the effect of temperature on development rate for one species. People would often, and I've done this too, set up a single rearing container for that species and that temperature, and then they would put that single rearing container in the incubator, and let's say you wanted to sample at 12 different ages. Then at each of those ages, you would pull the container out of the incubator, pull some larvae out, and put the container back in the incubator. Don't do that. I won't go into the details here, but that's not a good way to do this. And then when they talked about how they pulled out, 5 or 10 larvae from the container, they would often claim to not do that in a random way, or they do it in a biased way, namely pull out the largest larvae. And the claim was that this was analogous to what is done at the crime scene, where you do target the largest or most developed individuals. Well, we wanted to see what this would do if this were your sampling scheme. What would that do to your prediction? So I'm going to show you some complicated graphs here. So this is a simple rearing experiment, again, with Chrysomya megacephala. You're getting the idea that I like Chrysomya megacephala. Reared at one temperature, And these are a series of ages, and those samples were generated by there being a separate rearing container for each of those ages. We pulled it out, dumped every larva out, killed and measured every larva. The histograms on their side show the distribution of lengths for each age cohort. And then having all the data, we simulated subsamples. So on the left, you have an inverse prediction function, which is this red line. And I'm sorry if anybody's colorblind. So that's the statistical function right there. And on the left, that statistical function is made using 10 randomly sampled larvae for each age. And on the right, it's made using the 10 largest larvae from each cohort. And what we see here is we did predictions based on this cohort here, which is a 60-hour-old larvae. We took the measurements from each of those 60-hour-old larvae and predicted the age. And you get this line right here. And this is the coverage, which I talked about earlier. This is the measure of prediction performance. And remember, each of these predictions is an interval. And this line shows the portion of those predictions, I can't remember how many, it's about 70, included that particular X value. And the first thing you see is what you really want, which is the value is very high at the true age. And I can't see if it's 100%, but almost all of the predictions included the true age. And since these are 95% predictions, you would want it to be at least 0.95, and we certainly achieved that. So it has performance in that. What you also want is for this line to fall away quickly on either side of the true age, because that indicates you're not including in your predictions so many incorrect values. Now, there's no hard and fast objective standard for how quickly it should fall away. But if you, for example, were testing prediction performance based on two separate variables, and one of them had very narrow, steep coverage peak, and one of them had a very broad coverage peak, you would prefer the narrow peak, because you're sweeping up fewer incorrect values. And then if you haven't realized by now, prediction based on the largest larvae is terrible, right? Less than 20% of the predictions included the true age. So if you sample in that way, you're ending up with a prediction method that will likely be wrong. You'll incorrectly predict age. Typically, hurry up here, typically you have some sort of reason to choose that level of replication to give you a certain amount of statistical power. In the case of categorical data that I mentioned before, we have provided a table that tells you exactly how much replication at least the minimum you should need. So I'm going to show you a rather complicated table here, but this is the table that we provide in a couple of our publications. So it's unfamiliar, so I'll try to take it through you slowly, although we're pressed for time. So this is minimum sample size for, in this case, 5% significance. C is the number of categories. In my hypothetical example here, I'm talking about a development study in which you have 6 development categories. So egg, 3 larval instars, pupa, adult. And so for each age, you record the number of individuals in each of those categories. And you can potentially reject if your replication size is at least 37 to 83, that you have at least that many individuals for that age. You could reject if the training data had 0 individuals with that instar. If you have less than that, if you have 36, you can never reject. Okay, you cannot do these statistics if you have fewer than 36. If you have 84 to 136, then you could reject if it's 1 or 0. And there are reasons you would prefer to be to the right. I won't go into now. Notice right here, the upper left-hand corner, that's the minimum. So if you're down to, and this could be succession data as well, 2 categories would be like doing succession, looking at a single species and show presence or absence of that species. If you want to make 95% predictions and you have fewer than 7 pigs or humans, you can't do it. If it's 10% significance, in other words, you're making 90% confidence predictions, it's 4. It's fewer than 4, you cannot do it. And I can't tell you how many papers there are in the forensic entomological literature where people did a succession study and did 3. And how are we on time? I'm going to skip this part. No, I'll go through it real quickly. So people have often look at the effect of some covariate. Covariate would be like a tissue type that maggot's growing on. How does that affect growth rate? How does it affect a particular toxin? And usually what they'll do is some test of statistical significance, like analysis of variance, to see if there's a difference in growth rate according to the treatment, like what they're growing on. And they'll find some statistically significant difference and claim that this is important. But I believe you're asking the wrong question. What you want to know is that difference enough to make your prediction wrong. If tissue type, for example, affects growth rate, but it not enough for you to reach the wrong conclusion, then I don't think it's important. And so again, what we took here was we did a growth study. We priced my megacephalon, 2 types of tissue, pork heart, pork liver, just made some very simple growth curves. We tested the difference between those using analysis of variance, so age and food type, highly significant difference between the 2. And yet, if you took the data from the heart larvae and used them to predict the age of the liver larvae, you were right almost all the time. So in a practical sense, it doesn't matter. The difference between the 2 tissue types does not matter. Okay, so thank you for letting me go along. I'm just going to end on in case you were thinking that I'm casting doubt on all forensic entomology, I'm not by any means. Just show you that you can reach solid conclusions, even in very simple cases, and you don't need any statistics. So this is a case in Berkeley, California. There was a house fire reported early in the morning. It was quickly extinguished. A corpse was found in the house fire, sent to autopsy right away. Neither cause or time of death could be determined. The skull was fractured, but a fire will do that. The pathologist, and there was a suspect. There's somebody who had motive. And that person claimed to have spoken to the victim at 10 p.m. the previous evening. Oh, she was alive at 10 p.m. I talked to her on the phone. Pathologist, not on purpose, collected some larvae along with some tissue samples. You may not be able to see that larva at the bottom is 15 millimeters long. Do you know what you're looking at here? What do you see? Therian star. On the right is a blowfly. On the left is a flesh fly, a sarcophagid. That individual there is a 15 millimeter sarcophagid larvae. So if the suspect was telling the truth and the person was alive at 10 p.m. the previous night, that larva developed between 10 p.m. the previous night and 12 hours later. And no way, I don't even have any data on this, but there's no way that that larva developed that quickly. The person was lying. The suspect was lying about the person being alive. And in this case, the suspect actually confessed in court, presented with this evidence, confessed in court. Okay, so sorry for going a bit long. I'd like to acknowledge my longtime collaborator, Dr. Lamott, the wonderful people in my laboratory, some shown here, some not, and financial support from the National Institute of Justice. And thank you all for listening.
Thank you very much, Dr. Wells, for your excellent presentation. Now we are in Q&A session. So for participants who are attending the talk online, please, you can put your questions in the chat box and I will read a question for you. Alternatively, you can also switch on your microphone and ask questions directly to our speaker. And for our participants the physically attended this talk, please hold your question first. We would like to give opportunity to our online participant first. All right, may I know, is there any question from our participants online? Right, is there any questions?
Hi, I have one.
Hi, Moraya. Yes, please.
Yeah, when it comes to decos, and the ones especially that are found in water, as well as have been exacerbated by some type of heat, so like died in front of a heating source, how do you still measure time of death? Would heat affect any of the insects that you're found on the body?
So you're asking, how does one account for temperature in your analysis?
I'm sorry, can you repeat that? You went out. Say that one more time.
Are you asking how does one typically account for temperature when estimating time since death?
Yes.
So in entomology, there is an index that includes both time and temperature. So if you're talking about insects and you're estimating the age of the insect, what you're doing is not estimating age in time, you're estimating age in thermal units. I'm skipping a lot of details, but there's a standard way to do this. Now, what this has to do with, there's nothing particular about aquatic systems. Aquatic systems are rather difficult because in an entomological case, because there are no carrion specialists. So it's as if you were in a terrestrial environment and only dealing with ants and cockroaches. But the general principle concerning temperature is the same. So there is a standard way to account for it.
Thank you.
All right. May I invite another question? If there is any.
Hi.
Yes. Yeah.
I'm Rohayu. I just want to ask Prof regarding, you were saying that, the length of the maggot is not important, or it does not actually affect the postmortem interval because there are many variants that can affect the length of the maggots. So the length of the maggot, it does not really have any effect or contribution to the postmortem interval? That's my questions.
So in the example I gave, length was not valuable if you measured width. So in terms of measuring size, width was more valuable than length.
Okay.
And you have touched upon the reason. Maggots are soft, and length is a function of how it was preserved. That's why we produced a paper on instar. A lot of people don't use size because they believe it's affected by preservation method.
Okay.
But instar is objective. Preservation method doesn't change instar. But again, in the example I gave, it wasn't that length was useless. It was that length didn't help if you measured width.
Okay.
Does that make sense?
Yeah. You mean the width actually, like you don't measure the length because of the preservation technique, but you measure the width, the one that will contribute to the postmortem interval, is it?
In the example in our data,
Okay. All right.
width was more valuable.
All right. Okay. Thank you very much.
Okay, thanks, Dr. Rohayu, for the question. Is there any more questions from the online participants?
Hi, Dr. Hill, I have one.
Hi, Dr. Sean Lee from National University of Singapore. Yeah, please.
Hi, Prof. Wells. Thank you for your talk. I have just one question with regards to how you would frame a conclusion. So, for example, for one of your slides, you talked about feeding using pork heart and pork liver, and you mentioned that the results are pretty similar. So how would you frame a conclusion like this with regards to whether there is a significant difference between both types of meats for fly feeding?
So my point there was that people are concerned with the effect of various environmental conditions on maggot growth. And this concerns the task of selecting reference data that are suitable for your crime scene. So very often a laboratory growth experiment will have been done with meat from the grocery store. So pork liver or something like that. Very seldom done with human tissue. But even if that doesn't matter, let's say you reared your larvae on liver. Liver is cheap. Liver is also uniform, and people like that. There are reasons to like it for growth experiments. But you have larvae from the corpse that are not from liver. They weren't they were on head or muscle, brain or muscle. So people wonder, is data, our data from larvae grown on liver appropriate for estimating the age of a larva that developed on, say, brain? So that leads you to ask the question, is growth rate the same on brain as it is on liver? So people will do an experiment and they'll rear larvae on brain and they'll rear larvae on liver. And then they'll typically do some comparison. What is the effect of tissue type by like an analysis of variance? And they'll often reach the conclusion that they're different, that the growth rate is different on brain compared to liver. And the thing about these insect experiments, your sample sizes are so high that if there is even a tiny difference in the growth rate between those two tissue types, that will show up in your analysis and it'll be statistically significant.
Oh.
What I am suggesting is that that's not the right way to analyze the data. What you want to know is if you take growth rate data from liver and predict the age of larvae that developed on brain, even though there is an effect of tissue type, would you be wrong in your estimate of age of the larva? That's what's important. Will you be wrong or correct when you estimate the age of larvae grown on one tissue type using reference data from larvae grown on another type? And so I'm suggesting that that is how you should do the analysis. If you are concerned about the effect of tissue type, you should make a prediction function from the larvae grown on one type and see how well it performs predicting the age of larvae on the other type, because that's the practical implication. And in our simple experiment that I presented, we grew the same species, Chrysomimesis at the same temperature, 27 degrees, on pork heart and on pork liver. Those growth curves were slightly different, but the numbers were so high that we detected a highly significant difference using analysis of variance. But if you took the data from one tissue type and used it to predict the age of the larvae that grew on the other tissue type, you were very accurate in your estimates. So in that simple example, tissue type didn't matter. You didn't need to worry about tissue type because you would not be wrong if you took data from one tissue type and applied it to another. So I'm suggesting that that's the way to approach these kinds of questions. There are many investigations in the forensic entomological literature where people look at the effect of tissue type, of drug concentration, of pesticide concentration, things like that, and see whether there's an important effect. And I'm suggesting that the way you should test if there's an important effect should be through prediction performance, not some like comparison of means or something like that. Does that make sense? Right?
Yes, I'll go and take a closer look at the paper. Thank you.
Okay.
Okay, thank you, Dr. Sean Lee. Due to time constraint, we will allow one more last question from the audience. Last question from the audience. Any last question? Three, two, one. No more question? All right.
Thank you very much.
Yeah. So yeah, thank you, Dr. Wells, for your presentation. On behalf of the IMMB, Faculty of Medicine, UITM, we would like to thank Dr. Wells for your time and sharing of your experience with us. We highly appreciate it. I'm sure our faculty members and all participants today have learned something new and have been inspired by your stories. With that said, I'd like to thank all of you who are with us today, and please fill up the attendance form for the certificate. Thank you, everyone, and stay safe, take care, and I see you again. Thank you very much. Bye, everyone. Bye. Thanks for coming, Dr. Kurahashi. Yeah.